Good afternoon. My name is Dr. Laura Strawn, and I'm an associate professor and extension specialist at Virginia Tech. And I am uh, very happy to be here with you all virtually at this year's SARE Conference 2020 to present my work, Factors Affecting Prediction of Pathogen Hotspots. We'll start with a little bit of an overview of GIS, or Geographic Information Systems, make sure we're on the same page. And then we're going to talk about two particular studies where we've started to use this in produce food safety to predict pathogens. And we'll focus in on Listeria monocytogenes. And then we'll end sort of with our final thoughts and where we see this going. Geographic Information Systems is a computer system that is used for storing, capturing, checking, and displaying data related to positions on Earth's surface. GIS can also show many different types of data on one map. And this enables us to be able to easily see, analyze, and understand patterns and relationships. And when we're thinking about food safety, and specifically in pathogens in an environment, it's all about seeing those relationships and those patterns to predict where are those pathogens going to be, since our working hypothesis is that pathogen prevalence is not uniform in the environment. We know that there are pathogens in the environment, but we figure that there are certain times of year and certain locations where they'll be more prevalent than others. And once we can identify those, that'll help us with food safety mitigations. So with GIS, it supports various data, both remotely sensed from satellites and radar. A lot of it is publicly available. We can input census data, surveys, and we can analyze multiple data sets and develop algorithms that we can use for predictions. And so the graphic to the right shows you street data, building data, vegetation data, and it combines those all into an integrated visual where you can actually be able to look at complex interactions and sort of this 3D view of the world. I think it's worth noting one of the first applications of GIS was actually used in public health. John Snow, we're going to go all the way back to the London-based physician, 1854. They were having a large cholera outbreak, and he was able to take and demonstrate spatial associations between cholera cases and a single water supply. From there, once they identified this single water supply, they were able then to eliminate access to this water supply, ceasing the outbreak. And so back in John Snow's day, they took cholera deaths. These are the red dots on one of the graphs. All the blue dots are the pumps. And you were able pretty quickly visually to see that all of the cholera cases were surrounding the Broad Street pump. In today's world, we can use contour plots and mapping software to visualize these spatial associations. We have an intensity plot, and you can easily see that the Broad Street pump is where all of the cases are clustering, or the highest intensity in red. From there, eliminate the Broad Street pump for your water, and outbreak solved. And so this is sort of how we can utilize some of these spatial associations to be able to predict what's causing a problem, in this case, the source of the outbreak, and how we can refine our mitigations. So let's look at these in two examples in produce food safety. One, our first study, we wanted to look at the prevalence, persistence, and diversity of foodborne pathogens in the produce pre-harvest environment. And then we wanted to start and utilize some of these prediction model builders to identify factors, whether they be landscape features, soil characteristics, or meteorological events, and really predict a higher prevalence of where we would see this pathogen. We're going to focus in on Listeria monocytogenes today. It's a perfect one to use. We can find it. We know it's in the environment. So now our goal is it uniformly spread across the environment, or can we actually use predictions based on these factors to pinpoint where it'll be higher or lower? using this, we took a combination of field studies and computers. We did a large environmental sampling effort. You can see pictures where we took the date of when we sampled, the location, and we took drag swabs, which are basically a good capture of the topsoil. We took soil samples to get underneath what that microclimate is like. We took water samples. We were able to pull remotely sensed data. So you can pull a host of weather data with the date, with that location. You can pull landscape features, how close or far away are you from certain things. And then also we can pull characteristics on the soil. And then we can do some statistical modeling to kind of figure out what's going on, right? How can we use this for prediction? So one of the first things that we did was we built these classification trees based on high and low Listeria monocytogenes prevalence. So overall in the whole study, we found 15% prevalence of Listeria monocytogenes. Again, this is not to be unexpected. We know we can find Listeria in the environment, especially if we go out looking for it. What we wanted to know is, could we actually figure out areas where it was higher or lower based on certain factors? So through our computer modeling, we were able to generate a list of factors that were able to parse out high and low prevalence locations or times of year. So our first thing where it divided basically a high prevalence or a low prevalence was based on your location to water. If you were close to water, you had about a 40% chance, 39% chance prevalence of Listeria monocytogenes, versus if you were far away from water, that predicted prevalence dropped down to about 12%. You can also see that we started to make associations based on weather. So if it was cooler, you were able to find more Listeria monocytogenes. And then one of the interesting things is it started to break it out based on proximity to certain landscape features like urban proximity, whether or not you were around more cities or impervious cover, right? Roads, concrete structures. We were able to see that you could have, if you were close to those types of urban factors or features, you had a predicted prevalence of 20%, versus if you were farther away, only 10 meters, 5%. So not very far, right? 10 meters, it's 30 feet. So you had to be pretty close, right? So where you see some of these fields butt up right to urban features. We are also able to see soil available water storage and soils. So we started to get our first soil characteristic where we saw if it was a higher available water storage, we were able to get 30% predicted prevalence versus 10%. And then we were able to see the pasture proximity. If you were close to pastures, about 50%. And then if you were far away from pastures, 7.5%. And so we really wanted to be able to validate these models and figure out, would this work, right? We did this research study. We came out with our factors. Let's try to apply this to commercial ag. So we enrolled some commercial growers who were kind enough to let us take their farm fields. They were able to give us maps of their fields and tell us where they owned land. And we were able to build these predictive risk maps based on the different features that we had identified in our models, which was close to water, close to developed areas, low or high available soil moisture, and close to pastures. And what you can see, is that our predictive risk map shows that there's going to be different risk levels, higher predicted prevalence areas versus lower, which is what we hope to see, that you don't have this uniform risk. And so we wanted to go out and then validate it. Well, we went out and took samples in all of the higher areas versus the lower areas to see if we could, in fact, validate these models. And we were able to show that, yes, areas that we thought, for example, this little blue area of this particular farm, not a very large area, but here we were able to find significantly more Listeria monocytogenes than, say, other areas that were predicted to have a much lower prevalence of Listeria monocytogenes. So this was really exciting for us to see that, at least in the Northeast, where we had some of this data, we were able to start to get these predictions that were showing up correct. This also allows us where farmers can then decide and implement different mitigations, right? Based on, whether or not we can do different, plant different crops there, whether we can do different rotations. Is there a way where we can, try and do better laser leveling of fields to reduce some of that pooling of water or whatnot? Various different mitigations, and all of this is really tailored specifically to what you see. So our second study, we really wanted to kind of hone in on some of these weather and landscape features. We wanted to continue validation of our prediction to be able to see if we could use this. Here, we went away from commercial farms because we really wanted to control a lot of the different things that were going on. So we took a research farm where different weed scientists, plant pathologists, soil fertility experts are planting crops. They're trying to do this as much in the way that commercial farms would be run for research purposes. And we were able to look at two different fields. We figured through our models that one of our fields, A, would have a higher predicted prevalence because it was closer to water. It was down towards the southern end of our farm, where you can see in our predictive risk maps, looking towards the southern area of this southern part of this farm, where it's red and green. This is where, red and orange. This is where our models, based on the different landscape and soil features, were pinpointing higher predicted prevalence versus B, which was up towards the north part of our field where it was blue. So we wanted to look and see what we found in terms of Listeria monocytogenes in these two spinach fields from planting to harvest. We wanted to identify factors, see if we could actually validate those prior models. Were we actually seeing what was correct? And we also wanted to look specifically at some of our weather factors, and as well, did we see a transfer to the produce, right? That's a huge question, right? We can certainly tell you where Listeria monocytogenes is going to be in the soil, but would that actually transfer to the crop? So looking at this, we saw a pretty interesting distribution in our fields based on soil drag and leaf samples. We were able to see from our water samples that about 80% were positive for Listeria monocytogenes, which made this a really unique experiment since we were actually irrigating from that creek. What was interesting on this is that we assumed that the drag swab samples would be higher because they covered more surface area. But what we thought was really promising is that our leaf sample prevalence was very low. So we didn't see a lot of Listeria monocytogenes on our leaves, which showed that the transfer is quite low, less than 2% in this particular study, which was really great when you're starting to see sometimes prevalence is as high as 20%. When we looked at sort of what risk factors were influencing Listeria monocytogenes from being detected in these fields, we had noted that field A from our models would be higher in predicted prevalence. And we were actually able to see that field A was 2 times as likely as field B to find a Listeria monocytogenes positive. We were also able to see that rainfall within 3 days, we were 1.5 times more likely to find Listeria monocytogenes within a rainfall event. So this was nice because we were able to validate some of our model predictions in terms of picking out those fields or those locations that would be at higher risk. And then interestingly, we were able to see rainfall was really making a big difference. When we looked at the rainfall more specifically, here we have the predicted probability of Listeria monocytogenes, and you can see the more rainfall that you were getting, the higher predicted probability. And this mirrored in what we were seeing in our actual field data as well. Looking at field A specifically, this was our higher likelihood of finding Listeria monocytogenes. We really wanted to see were we seeing this again, where we saw spatial clustering or where it wasn't uniform. We were able to use some of these Ripley's K functions to show that we were getting the yellow-blue areas They indicate Listeria density. So hotspots are in the green and yellow, which are around the edges, not in the center. We sampled these plots every 5 meters. So we had a really good distribution or uniform distribution of sampling. So if it was uniform in the field, we would expect to see a little bit more of a random pattern, but we weren't seeing that. We did a quadrant analysis, and this also through some conditional Monte Carlo simulations was able to show quite significantly that this result indicated evidence of Listeria monocytogenes. Positive soil samples were clustering in field A towards the edge. So we had edge effects that were going on. It was not random. Looking at our distribution of Listeria isolates, this also mirrored the same thing. We have our GIS-based image. We have our hotspot density plot. And again, we're able to see that on that edge, we're seeing all of those different Listeria pop up. We're actually also able to see, too, that the red dots share the same allelic type as what was coming from the creek. So somehow, even though we were distributing, this irrigation water uniformly in a field, we're getting these edge effects, whether that's based on low levels of the field or drainage. This is where we're starting to see the Listeria hotspots form, is towards these edges of this field. So kind of our summary and final thought is that these GIS-based models show a lot of promise in identifying trends and establishing measures of risk associated with contamination events. What was nice to see is that we were able, based on sampling efforts and prediction models, to go out into fields, whether commercial or research farms, and show that those predictions were actually working. When we thought we would find more Listeria, we were, based on the computer models. And this is nice because it might yield a big return on an investment for food safety. You're going to have gaps that encompass the overall farm. But if you know that there's particular fields or particular locations on your farm where you may have a higher likelihood of Listeria monocytogenes or a pathogen, this is going to cause you to change some of your things. Can you do different crop rotations? Can you avoid that field at certain times of the year? Can you put in different mitigations, whether that be planting an extra row or two that you're not going to harvest near those edges, whether this can tie into laser leveling of fields for better drainage. There's many things to do if you can target an area or a problem area, especially if you can forecast different knowledge. Of course, it always takes a village. I'd like to thank my colleagues at Virginia Tech, Cornell, and Florida, as well as my laboratory group. I'd like to thank the funding sources, and a special thanks to the produce growers who participated in these studies. They were able to allow us and give us a lot of trust and freedom. And hopefully in the end, it was really interesting for them to find out sort of where on their fields, if a lot of them had ideas of where they thought there might be more or less pathogens based on certain knowledges they had of the field, where the animal intrusion is, where low levels of the fields are, et cetera, different soil types, and seeing how these kind of interplayed with the models was really fun. So with that, thank you, and I'll take any questions.